around the same time that Griffith was doing his transformation principle experiment with mice, Avery, McCarty, and MacLoed found out that you could actually do it without using animals, simply by growing the bacteria in culture and then looking at the changes that you see within the strains that are present in that culture. So first, if you put your living R strain along with heat-killed components of your S strain together in a tube with culture, and then you let them grow over time, what's going to happen is that there's going to be this transformation, and then you're going to end up with just living S strain. So something was causing this transformation, but at the time we didn't know what it was. And Avery decided to call this substance that was causing the transformation principle. And he and his team worked on this for many years and finally isolated this transformation principle substance, which I drew in this red test tube over here. And then they, although they had this like pure sample of this transformation principle, which we now know is DNA, they needed to find out what it was actually made of. So the way they did this was through a series of experiments using different enzymes, breaking down different components of what could possibly be in this principle. Okay, so Avery and his team took this transformation principle and exposed it to a bunch of different enzymes. So the first one was protease, which breaks down proteins. So if the proteins are broken down, they're not going to be able to do anything, and therefore they cannot cause a transformation. So if we see a transformation happen, and we did, the R strain, when you added this purified transformation principle along with the protease in it, and you added that to the R strain was still transformed and turned into the S strain. Now, if proteins were responsible for the transformation principle, we wouldn't have seen a transformation because proteins aren't working here anymore because they're broken down. Now, same thing with RNA. If it was RNA, well, we used RNAs and we broke down RNA, and then we mixed up this transformation principle along with the living R strain, we still got a transformation because we ended up with S strain. Now, the interesting part is when you get to DNA. So if you use DNase, breaks down the DNA in your sample, and then you add that to the living R strain, you don't see a transformation. You still have R strain after. So the only time that we didn't see a transformation is because the DNA was not acting. The DNA was not able to do its work and actually cause this transformation. Even, checked if it could have been fats in the sample or something. So he eliminated the fats through ultracentrifugation and still saw a transformation because fats are also not responsible for the transformation principle. And then further physical and chemical analysis showed that it was that the substance that they were looking at was actually DNA. And yet, despite all of this evidence, the scientific community was like, no, you probably messed something up. Maybe there's some sort of contamination, or maybe it's not actually a genetic thing that's causing this transformation. Maybe it's just some other physiological thing going on. I don't know. They just said, it's probably still proteins. But then further experiments came along and said, hey guys, it's actually DNA, not protein.